Hello, everyone. We're glad you've joined us for this live webinar, A Complete Workflow for Analysis of cfDNA from Plasma to Variants. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Beckman Coulter and Integrated DNA Technologies. To learn more about our sponsors, please visit them at www.beckman.com and idtdna.com. So let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. So to do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of your presentation window. If you experience any technical problems seeing or hearing this presentation, just click on the Support tab found at the top right of the presentation window or report your problem by typing it into the question and answer box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process for obtaining those credits. Without further ado, I now present today's speakers. Shilpa Parakh, a Senior Application Scientist in Automation and Biotechnology Business Unit at Beckman Coulter Life Sciences, and Nicole Roseman, a Research Scientist at IDT. Welcome, Shilpa and Nicole. You may now begin your presentations. Welcome again.
Okay, thank you. In this webinar, we will review why liquid biopsies are challenging, what is conversion, NGS workflow and automation overview with Beckman's Apostle Minimax High Efficiency Isolation Kit, xGen Prism DNA Library Prep Kit, and xGen Hybridization Capture Reagents. We will demonstrate an integrated reagent workflow for cell-free DNA analysis, maximizing cell-free DNA recovery from low volumes of plasma, higher conversion and complexity, sensitive and reliable variant calling at less than 1% allele frequency, and high on-target rate. Liquid biopsies, such as a blood draw, are much less invasive than tissue biopsies. So if you see the person here on the left-hand side, theoretically, all metastatic sites are shedding into the bloodstream, representing the full tumor heterogeneity better than a localized biopsy sample. Cell-free DNA can provide highly personalized monitoring, including therapeutic, predictive, and prognostic information by being able to collect multiple samples over time. Why is conversion critical to liquid biopsy analysis? So conversion is critical because losses can occur at different points in the workflow. You see on the right-hand side in the dark blue, the workflow steps include poor recovery from plasma during extraction, low conversion and loss of coverage during library prep, loss of complexity and low on target during hide capture, and finally, errors that occur during PCR and sequencing. All of this can lead to a poor limit of detection. When looking for low frequency variants, limit of detection is important in accurate identification. Low yield and losses throughout the workflow decrease confidence in the data, consume precious sample, and may require repeating the prep. Why is conversion important? So let's start here with at the very top, you have your input material, your sample. You have your variant of interest in the dark gray that has a little red nick in it. This is like searching for a needle in a haystack. You need all of your molecules and cannot afford loss. So if you go over to the right arrow for low conversion gives you a library with lower complexity. you may lose your variant of interest, as you see with no dark gray with red nicks. And then moving down again with the arrow for low coverage, low library complexity also results in fewer reads, mapping to that region. Lower coverage decreases sensitivity. So following your sample with high conversion on the left, the dark gray or the dark blue arrow, whereas when you have high conversion giving you high library complexity, you retain your variant of interest, as you see in the dark gray with the red nick. And going down to higher coverage, not only have you captured your variant, but you also have higher coverage and thus higher sensitivity. Conversion is critical for somatic mutation detection. Clinical samples are often challenging because not only are they invasive, but they result in small amounts of DNA, where cell-free DNA is limited to maybe only a few 1,000 genomic equivalents. So looking in at this table, on the left-hand side, you see that large pieces of tissue samples with resections and excisional biopsies have a high success rate in target enrichment test. And this column is labeled test completed successfully with the percentage. However, you see samples drop below this 50% success rate from core biopsies and fine needle aspiration due to not being able to get enough DNA. If the tissue heterogeneity does not have the variant you want, going back to the patient can be painful. Cell-free DNA through liquid biopsies is less painful, and if you can increase conversion, you can increase your success rate. So Shilpa here will discuss how to address the conversion challenge.
Thanks, Nicole, and thank you all from Beckman Coulter and IDT for attending the webinar today. So we just heard from Nicole why conversion is important. In this segment, we are going to see how we are addressing this challenge. This workflow is our approach to address this challenge. The samples are extracted using Apostle Minimax high-efficiency DNA isolation technology by Beckman Coulter, followed by library prep by IDT's newly launched Prism DNA kit. The libraries are then taken through IDT's hybridization and capture workflow and then sequenced on Illumina's NextSeq platform. cfDNA is of clinical relevance as it helps in early detection of cancer or tumor regression. Apostle's high-efficiency cfDNA kit can be used to extract DNA from large volumes of samples like plasma, serum, urine, etc. For the automated application, it can extract up to from 4 mils of sample and manually from a maximum of 5 mils. The kit is compatible with several blood collection tubes. It supports flexible input and is automation friendly. Another very important benefit is Apostle Minimax efficiently removes PCR inhibitors to allow for use in NGS and PCR-based assays. This data is from qPCR assay and shows the yields for low and high input volumes between different kits. At low inputs, the yields are comparable, but at high volumes, Apostle's yields are higher than the other kits. This is a bioanalyzer trace with Agilent high sensitivity DNA chip for replicates from Apostle and another kit, and the traces show higher yield for cfDNA extracted from Apostle kit. Figure on the top right shows a comparison of yields from samples collected in different kinds of tubes. 3 different tubes were tested, the EDTA tube, Streck, and PaxGene tube. The yields are higher with Apostle for all the 3 tube types. Kit A still has some yields, but lower than Apostle extracted cfDNA. But Kit B did not have any yield from non-EDTA tubes. The last figure represents the comparison of PCR inhibition. Undiluted samples were compared to samples diluted in 1:4 ratio to measure the effect of PCR inhibitors. If PCR inhibitors are present, then the estimated concentration will be higher in more dilute samples. And if there are comparable concentrations from dilute and non-dilute, then it's a sign of low inhibition. As you can see, Apostle Minimax and Supplier 1 have low inhibition, and significant PCR inhibition is seen in Supplier 2. In the left image, blood samples were drawn from 4 healthy donors in K2 EDTA tubes. The plasma was harvested by centrifugation, and cfDNA was extracted using Apostle kit and Supplier's A cfDNA kit. The cfDNA yields were assayed by qPCR. Apostle yielded about 25% more cfDNA than Supplier A. It indicates that Apostle Minimax has consistently higher extraction performance. In the figure on the right, it's a tape station trace from donor B. cfDNA size distribution was analyzed on Agilent high-sensitivity D5000 screen tape. cfDNA extracted from Apostle Minimax has typical mononucleosome peaks at 175 base pair and dinucleosome peak at the 350 base pair mark, with a high efficiency compared to Supplier A. Clearly, the cfDNA quality and performance is better for Apostle Minimax kit. With that, I'm handing it back to Nicole to take you through IDT's case study.
Thank you, Shilpa. For IDT's xGen Prism DNA library kit, let's just dive into that. It provides sensitive and accurate variant detection from degraded samples such as cell-free DNA and FFPE. xGen Prism gets higher conversion rates compared to TA ligation-based methods through use of an engineered ligase and highly modified adapters. These adapters have a single concentration regardless of input type or mass, creating an easy workflow. You can get data from even the most degraded samples and detect variants at less than or equal to 1% variant allele frequency from libraries with higher yield and complexity. This minimizes your sequencing errors with unique molecular indexes and minimize sample misassignment with unique dual indexing primers. Traditional TA ligation library prep kits are less efficient than blunt ligations. However, they prevent adapter dimers, and as you see in the box to the left, they also prevent insert-to-insert ligation chimeras. Adapter dimers are full-length adapters that can bind and are able to cluster on flow cells during sequencing, and this is shown with the green and light blue. Let's walk through the mechanism of IDT's xGen Prism DNA library prep kit. At the beginning, input DNA should be fragmented with mechanical shearing, such as via a Covara system. Cell-free DNA is already fragmented to the appropriate size. The end repair enzyme mix converts input DNA into blunt-ended DNA ready for ligation, which you see with below the arrow. Next, the ligation 1 enzyme catalyzes the single-stranded addition of the ligation 1 adapter to the 3' ends of the insert. The 3' end of the L1 adapter contains a blocking group to prevent adapter dimer formation, which you see here in the picture. There's a little red circle with an X through it. This also allows for a fixed concentration of adapter and no longer requires a size selection, producing higher conversion and library complexity when compared to traditional TA ligation library prep methods. Additionally, 6 single-stranded UMI sequences are added to the insert, which you see in dark blue and light green, and they're labeled with a UMI. This unique single-stranded ligation to fixed UMIs enables strand-specific molecular indexing by independently tagging the top and bottom strands. The ligation 2 adapter acts as a primer to gap-fill the bases complementary to the UMI, which you see in the dark blue and lime green arrows going inward towards the insert. This is followed by ligation at the 5' end of the DNA insert to create a double-stranded product ready for PCR amplification. PCR incorporates sample index sequences. Our xGen unique dual indexing primer pairs minimize sample misassignment. After xGen Prism libraries have been cleaned up and QC'd, we take 500 nanograms of library into target capture. xGen blocking oligos are added and bind to library adapter sequences to reduce the off-target being captured and prevent daisy chaining. You see here the blocking oligos are the dark blue in the middle fragment. Probes, which are the light blue, hybridize to your target of interest. Probes are 5' biotinylated and pulled down via streptavidin beads. IDT offers two different types of panels. The first are xGen lockdown probes. These are individually synthesized, QC'd, and normalized prior to pooling. They're also customizable and modular. The second are NGS discovery pools, which have the fastest turnaround time. For less than 50,000 probes, the turnaround time is 5 days. These have high fidelity and are also individually synthesized. IDT synthesis and chemistry gets you the on-target coverage and uniformity during ultra-deep sequencing, which I will show data on in a subsequent slide. So going into an experiment that we conducted, cell-free DNA conversion was evaluated using Beckman's Apostle minimax extraction kit. And it was compared across 3 different library prep kits. Ours, the xGen Prism, Kit A, and Kit B. All of the libraries generated were captured and sequenced using the same method. A key performance metric to evaluate library prep is ligation efficiency. So taking a look at the graph, on the x-axis you have your yield input into library prep, and then on the y-axis is your ligation efficiency. IDT's ligation strategy is more efficient than traditional TA ligation-based methods. For 100 nanogram of genomic DNA input, xGen Prism has approximately 2 times the ligation efficiency, while 25 nanogram input is approximately 3 times higher than Kit A and Kit B. Ligation efficiency was measured via qPCR after ligation 2. Ligation efficiency is just one part of conversion. Stringent bead cleanups, PCR efficiency, and hybrid capture all affect conversion. So xGen Prism DNA library prep kits have higher conversion and complexity with cell-free DNA. So looking at the graph on the left, the x-axis is the library prep kit, and on the y-axis is conversion rate measured after sequencing. And all the reads were normalized to be comparable between the 3 library prep kits. xGen Prism libraries have approximately 3 times higher conversion when compared to Kit A and Kit B with 25 nanograms input of cell-free DNA. Moving to the graph on the right, again you see the library prep kit on the x-axis, and on the y-axis are the unique molecules measured after sequencing. xGen Prism libraries have approximately 3 times higher the number of unique molecules than Kit A or Kit B. Now I have data from a case study with cell-free DNA from healthy donors. So libraries here were generated according to each manufacturer's protocol using 25 nanograms of cell-free DNA. So looking at the infographic cartoon on the left-hand side, samples A and B were drawn from 2 healthy donors into K2 EDTA tubes. 4 mils of plasma was harvested by centrifugation, and cell-free DNA was extracted using the Apostle minimax high-efficiency isolation kit. Sample B was spiked into sample A to create an allele frequency of 0.25%. Libraries were then captured with a custom 61 kb xGen lockdown probe panel and analyzed with start-stop deduplication. Variants were called using Vardict or combined read family error correction. Sensitivity and positive predictive value were also measured. So using the extracted cell-free DNA, all things equal, input, same extraction, xGen Prism performs better than Kit A. The left graph compares library complexity, while the graph on the right compares start coverage. Again, sequencing reads were normalized between both library prep kits. Increasing your conversion increases your library complexity and thus your performance. Okay, high on target rates and uniformity, even from small capture panels, as our 61 kb xGen lockdown probe panel. Using the graph farthest on the left, you see that it gets high on target even for both library prep kits, xGen Prism and Kit A. However, moving on to the middle graph, it's comparing coverage using combined read families. On the y-axis is your target base fraction, and that's the percent of bases that get to at least that level of coverage. The graph farthest on the right compares coverage using start and stop. Here we demonstrate how efficient capture with high complex libraries from xGen Prism lead to higher coverage. Efficient captures have high on target rates and uniformity, and all of this enables a better ability to call variants. Digging into how we use UMI-based error correction and data analysis. So recall a fixed single-stranded UMI sequence was added to your library insert during ligation 1, which enables strand-specific molecular indexing by independently tagging the top and bottom strands. So one of 32 possible sequences on either side, which is demonstrated with the dark blue and lime green UMI label, give you enough diversity because the UMI sequences are fixed. Even if there are errors in the UMI, it is possible to identify and correct these errors, which prevent artificial inflation of library complexity. So comparing data analysis with versus without UMIs. First, let's look at analysis without UMIs. You have your original DNA fragment on the farthest left, and the little green circles are your true positive mutation. Moving through your workflow, you get errors introduced, such as through sequencing or through PCR. And the output is your sequencing data. Then moving on to the right, you get deduplication by start-stop position. And this goes with the highest quality read, and it might not contain your variant. So when you do analysis with UMIs, again, on the farthest left, looking at your original DNA fragments, the little green circles are your true positive mutation, but the colored ends are your UMIs, and they're added during xGen Prism DNA library preparation. Going through the workflow again, just like we did before, and the little red circles are your PCR or sequencing errors. So with the sequencing data, the shaded regions represent read families before consensus read generation. Using both the start-stop position and UMI sequence to deduplicate reads. This method uses all sequencing data from the read family to choose the most likely base at each position from beginning to end. This can be done with single read families or combined read families. So looking under error correction with UMIs and focusing on single read families, these use UMIs on one strand. You see one error still sneaks through. Then going over to combined read families, these use the UMIs on both strands, resulting in less false positives and increasing specificity. You need this high specificity and sensitivity for limit of detection, and this is done by our high conversion. After sequencing, reads were mapped using BWA, start-stop deduplication, or error correction across both strands were performed. So this is the same libraries from our case study that we went over, and it was captured with the custom 61 kb xGen lockdown probe panel. They reliably identify less than 1% variance with UMI-based error correction. So using UMI-based error correction dramatically increases variant calling accuracy while still maintaining great sensitivity percentage. This also greatly decreases the chances of false positive calls. So we have demonstrated an integrated workflow for cell-free DNA using a complete reagent workflow that guarantees high conversion. This maximizes cell-free DNA recovery from low volumes of plasma and obtains higher conversion and complexity using the xGen Prism DNA library prep kit, and produces sensitive and reliable variant calling at less than 1% allele frequency using built-in fixed UMIs. I'll now turn it back over to Shilpa to dive into automation.
Thanks, Nicole. As you saw, that NGS workflows are complex. So along with IDT, Beckman Coulter has designed a complete end-to-end automated solution to address customers' pain points, improve library quality, increase throughput, and faster turnaround time with least hands-on time are important components for successful results. Here, I'm showcasing the automated solutions for Apostle extraction kit, IDT's xGen Prism DNA library prep kit, and IDT's height capture kit. We are constantly trying to understand how to help customers in expediting their research by adapting to varied workflows and providing time and cost savings and quality results and support. Our solutions are flexible and scalable, easy to implement with support from our experienced applications team. Along with implementing recent innovations on hardware and software, the primary function of the Biomek workstations remains providing accurate and reproducible pipetting performance. We offer two sizes of I-Series automated liquid handling workstations with either 45 or 25 deck positions and 6 different pipetting configurations to meet your throughput and application needs. These workstations can be open or enclosed. We have 3 different multi-channel heads and different syringe sizes for span 8 probes. We listened to the feedback given by customers. These themes led to a common foundation across multiple applications for hardware, software, accessories, consumables, and support. The Biomek I-Series is simplified so you can focus more on the science and less on managing your tools with multicolor status light bar and light curtain for safety. It's efficient to help you optimize productivity, increase walkaway time with larger deck capacity, independent 360-degree grippers, and large multi-channel head. It's reliable to help maximize instrument uptime. Systems have 2 cameras and also come with Beckman Connect remote service support. And it's adaptable, so the technology you invest in can grow with you with grid-based design and standard device integrations. Applications developed at Beckman Coulter are demonstrated, which means they are written, optimized, tested with samples, and then they are ready to install at a customer site with faster implementation. Along with the great pipetting performance with the hardware, we also provide the user-friendly Biomek software with demonstrated methods interface tools. These are set of tools we utilize in these methods to provide ease of use for the operator to run complex workflows. So the DMI includes Biomek Method Launcher, Method Option Selector, Guided Labware Setup, and Deck Optics Final Check. Biomek Method Launcher software is designed specifically with the system operator in mind. The launcher presents the Biomek methods that are meant to be run by the operator from an app-style screen. Method Option Selector provides the runtime options to schedule the daily experiments. Guided Labware Setup is one of our advanced deck setup utility. It guides the user through deck setup by labware type and volumes with advanced support for labware stacking and reagent preparation. And Deck Optics Final Check identifies the problems before the start of the experiment. Further taking you through each of these tools, methods are run by Biomek Method Launcher to avoid the risk of editing the Biomek methods accidentally. The methods can be run from an app-style screen, and deck setup notes and master mix recipes can be printed to keep for your records. No need to navigate the core software as the system can be run from BML without unintentionally editing the Biomek methods. The Method Option Selector, or MOS, is an interactive user interface written keeping the modular workflow in mind with all of the recommended start-stop points in the IFU. Here I'm showing the user interface for all the three applications. For extraction, user can choose number of samples, input volume, elution volume, etc. The Prism library prep kit has sample number input, on-deck or off-deck thermocycling, on-deck normalization, and all the sections of the library prep. The method can be run as full walkaway or modular on i7. For hybridization, both dry down and Ampure bead-based concentration protocols are supported. Hype can be done on deck or off deck, and capture can be done on deck with the help of multiple Peltiers. Guided labware setup helps set the deck correctly and organizes details on labware and reagents, and leads you through your deck setup step by step. It provides detailed instructions for type of labware, its placement on the deck, and each reagent position and volumes based on the options chosen in MOS. On the right, you see the stock reagent tubes on the cold block. This is a modular reservoir with bulk reagents, and here's the final deck setup. The dynamic methods automatically adjust labware quantity and reagent volumes based on variable sample number. Human error is a part of science. There are a lot of components that get added to the deck, so it's easy to make mistakes. With the help of left and right cameras on Biomek, Deck Optics Final Check helps identify common errors that can result in wasting expensive chemistry, failed experiments, and delayed projects. Final Check ensures that you've placed your labware in the correct location and provides warnings if labware is missing or if the wrong tip type or plate types are detected on the deck. As in the example, you can see Deck Optics recognized that wrong tips are placed at 2 positions, thus saving the experiment. Moving into each application, Apostle Minimax is automation amenable and scalable. The market requires higher throughput extraction to expedite the downstream research, and automation plays a key role in achieving that target. Apostle Minimax cfDNA kit is automated on both Biomek i5 and i7 workstations. The i5 multi-channel has a throughput of 48 samples, which can be extracted in less than 5 hours. The i7 hybrid application is designed for a throughput of max 96 samples, extracted in a little over 5 hours for 4 mils of sample. Both the decks are equipped with orbital shaker, Peltiers, and incubator, and can extract from 1 to 4 mils of sample. The following is the data from Biomek i7 application. 4 mils of plasma was used to extract cfDNA for 3 technical reps of 7 different donors. 4 of the donors were from EDTA tubes and 3 were from xGen. On the left is comparison of qPCR traces for manual and automated extracted samples. On the right are the CT value comparison, and we can see they are pretty comparable with the manual workflow. We also compared the cfDNA yields for manual and automated samples via picogreen assay, and the yields are comparable or higher for automated workflow. The last one is a comparison of extracted cfDNA from different types of tubes. The blue traces are manual and red traces are for automated extracted cfDNA, and the results look quite comparable. This is the deck layout for IDT xGen Prism and IDT xGen Hype Capture automated solutions. The deck layout has been kept same for smooth transition between running both the applications. Each application supports a throughput of 96 samples with the help of ATC, wash stations, shaker, and Peltier. 1 Peltier helps keep the reagents cold, and the other 2 Peltiers help keep the reaction plate chilled during library prep or keep the buffers heated during capture. Normalization of sample is also supported on DEC. In the following figures, the white square is the DEC setup and the yellow time symbol is the recommended safe stop points. This is a flow chart showing how automated application is handled on i7. The automated protocol for Prism is a full walkaway solution once the DEC is set up using guided setup. But it is flexible to run modularly so users can plan their day. It starts at normalization and final library is ready after the PCR cleanup. This is the workflow for hybridization capture as automated on i7. After initial DEC setup, the application can run blocking and dry down or pure bead-based concentrating protocol. Hybridization can be done on DEC or off DEC from 4 hours to overnight. After Hybe, DEC is reset to replenish all the reagents and tips to carry on capture, PCR, and last cleanup. Here's a table to show instrument setup time and runtime for different sections of the protocol for Prism DNA library prep kit. The fully automated method can prepare 96 libraries in around 5 hours with only 20 minutes of hands-on time, whereas the hands-on time for manually preparing 24 libraries is around 2 hours. And on the right is hands-on time and turnaround times for Hybe capture protocol. For 96 captures using bead-based concentration and 4-hour hybridization, the captures can be done in less than 10 hours. The hands-on time is 3.5 hours for 32 samples prepared manually versus 45 minutes hands-on time to set the DEC for automation. We have several resources like application notes, overview flyers, and posters available for Apostle cfDNA extraction solution and IDT's xGen hybridization and capture, which cover the experimental design and data in detail and are available upon request. The xGen Prism DNA library prep project is currently in final stages of sample testing and will be available soon. To conclude, this is an end-to-end automated solution from extraction to captured libraries ready to be loaded on Illumina sequencers. Automation helps in reduce hands-on time and increase throughput. The higher throughput supports more barcodes and allows for multiplexing and full utilization of sequencers. The kits are automation-friendly and scalable, and automation supports bead-based technology for extraction and cleanups with enhanced selective tip pipetting for a faster turnaround. Demonstrated methods interface tools provide correct DEC setup and reduction in setup errors. With automation, the cross-contamination can be avoided and helps in accurate and precise sample transfers. The solutions are supportable by knowledgeable teams from IDT and Beckman Coulter. With that, we thank you again for attending this webinar.
And thank you, Shilpa and Nicole, for that presentation and for your important research. We will now start the live Q&A session of the webinar. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question and click that send button. Shilpa and Nicole will answer as many questions as time permits. So let's take a look at our questions coming in from our audience members. Shilpa and Nicole, our first question is, how are the UMIs being used in error correction?
I can take that question, and let me go back up to one of my previous slides since it has a nice infographic to explain this. Okay, so here is the infographic kind of showing how UMIs can be used for error correction. Remember here on the ends, the color, nice pretty colors, are they represent the UMIs. So UMIs are on single and on both strands, and they can be used to create single read families or combined read families. So consensus reads are then generated and aligned based on either single or combined read families. Rather than using the highest quality read, you are using all of your sequencing data within your read family, and this selects the most likely base at each position from beginning to end. So combined read families takes a step further and compares both strands.
Thank you for that. And what are the advantages of fixed UMIs over random UMIs?
Yes, that's a good question. Fixed UMIs enable strand-specific molecular indexing because they independently tag the top and bottom strands. So even if a PCR or sequencing error does occur within the UMI, it's possible to identify and correct for these errors, preventing artificial library complexity.
Thank you for that. And again, I want to thank our audience members for these great questions coming in. Our next question coming in, it says, how do indexing primers prevent sample misassignment?
So the experiment that I shared today uses unique dual indexing primers. So UDIs contain unique index sequencing or sequences for both the I5 and I7 index reads of the Illumina sequencing platform. So misassigned reads will be flagged as undetermined, and then they're excluded from analysis.
Thank you for that. And what are typical yields after apostle extraction from a tube of blood, for example, 4 millimeters of plasma?
This is Shilpa, and I can take this one. cfDNA concentrations actually vary significantly. They can range from 1 nanogram to 100 nanograms per mil of plasma. But the concentration typically seen for cfDNA for healthy donors is usually between 1 to 15 nanograms per mil of plasma.
Thank you, Shilpa. And we have time for a few more questions that are coming in. An audience member wants to know, can I use the Prism kit with TA ligation adapters or other IDT adapters?
So I know IDT does a lot of custom designs for people, but in this case, you cannot. So the adapters that are supplied in the xGen Prism kit have been optimized to enhance ligation efficiency. So these adapters in combination with our engineered ligase is why our conversion rates are so high. So, and then plus an added benefit is you don't have to mess with adjusting the concentration based on your sample input.
And during the presentation, you talked about the DEC optics software. What errors does that detect?
So once the DEC is set up using guided LabRoots setup, the dual cameras on Biomek will help using the DEC optics final check software, and that will detect wrong plate type or wrong tip box type, or if any labware is missing from that particular setup.
Thank you for that. We have time for one more question. There is gDNA contamination in a eluate of apostle minimax. How do you prevent that from happening?
So the improper handling of blood can result in gDNA contamination. Delay in processing of these tubes, especially in the EDTA tubes, will cause cell lysis and gDNA releasing into plasma. The incomplete removal of these cellular components can also result in gDNA contamination. And frozen whole blood can also cause significant cell breakage and gDNA contamination. So if avoided these steps, then the gDNA contamination will be less in the sample.
Thank you again, Shilpa and Nicole, for your presentation. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. As a reminder, any questions we do not have time for today will be addressed by the speaker via the contact information you provided at the time of the registration. I'd also like to thank LabRoots and our sponsor, Beckman Coulter Life Sciences and Integrated DNA Technologies, for underwriting today's educational webcast. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, Bye-bye.